Hello everyone, my name is Raul Mendez-Giraldez. I work at the National Institute of Environmental Health Sciences. Today I'm going to summarize my poster at the ISMB 2022 in Madison entitled Tree-Based Integration and Visualization of Single-Cell Data from Multiple Patients at an Early Stage of Acute Myeloid Leukemia Treatment. The prognosis of most of patients with acute myeloid leukemia is generally poor because frequent relapse, which is thought to be from the persistence of leukemia initiating stem cells following treatment. Early changes at the molecular level suggest a potential establishment of chemotherapy resistance that can mediate long-term survival. However, intratumor and intertumor heterogeneity confounds our ability to efficiently map cell lineages and state transitions during tumor progression and treatment response. Single-cell technologies such as cytometry by time of flight combine the platforms of conventional flow cytometry with mass spectrometry to analyze millions of cells simultaneously. For this project, our collaborators collected peripheral blood samples for 32 patients, 13 female, 19 male, processed into 7 batches independently, each of them with a healthy donor as a control. In the box plot below, you can see the distribution for the age of the female and male groups. The female group has a median age of 50 years, the male 64, but the female group has more spread. For each individual, the blood sample was run against a panel of 36 markers, 21 surface markers, and 15 intracellular markers that are listed here in this table. We have recently published a method to deal with time course data from single-cell experiments called dynamic spanning forex mixtures. It uses decision trees to select features that account for multimodality, skewness, and time variation. Those features are subsequently used to build the forest using tree agglomerative hierarchical clustering and dynamic branch cutting. For this mix to run, we need to input 3 different types of data. One profile matrix with the genomic features as a rows and columns the cells, a vector annotating the cell clusters or cell types, and then another vector annotating the time states for the same cells. In order to provide cell states, we use a combined approach that includes hierarchical clustering and conditional classification to yield the unique clusters across times for a given patient. On the other hand, we provide labels to those clusters by starting with the reference controls and then scoring each group of cells against the known signatures for peripheral blood published in the literature, and assigning the signature for a given group of cells that has the highest cosine similarity while being significant above the 0.95 confident level. As an example for DSF mixed results, we have patient 47, 1 AML 82. We show here 2 forests which are the same topology, just colored by time state on the left and by cell state on the right. The one by the time states allows us to distinguish myeloid versus lymphoid lineages. The myeloid lineages are dominated by cells at time equal 4 hours in the dark blue, and then at time equal 24 in light blue, states 2 and 3 respectively. Whereas the lymphoid lineages are in the beginning with the cells at time equals 0, and then all the rest are cells from time equal t equal 4 hours, states 4 and 5 respectively. If we compare with this forest color by cell states, we can see that the subtree corresponding to the myeloid lineages is made of monocytes in yellow, then basophiles blue, plasmacytoid dendritic cells in red, granulocytes in the turquoise, and neutrophiles in light orange. Whereas the lineages, lymphoid lineages are made from at t equal 4 CD4 T cells in this kind of blue, and then NK T cells in green, and then NK cells in orange that separated at t equal 4 from the B cells at t equals 0. However, it's difficult to build a time course trajectory. Another example of DSMX results are this one's for patient 47, 1 AML 53. You can see the two subtrees by time state. This is a subtree corresponding to the myeloid lineages, and it will be for the lymphoid lineage. Myeloid lineages is dominated by cells collected at t equals 0 in dark blue, and then some branches with t equal 24. However, most of them are in both cases monocytes, like in dark orange here, with some basophiles here. The tree for the lymphoid lineages has a nice separation between the B cells at t equal 0 versus t equal 4 and 5, which are made of CD4 T cells and NK T cells. As a conclusion, I would like to say that our framework deals with missing values at the selection of the cell and time states, as well as in the final spanning forest computation. It is scalable, capable to deal with tens of millions of cells, and hence can be generalized to other single-cell data modalities across multiple patients. The integration of all the patients in a single analysis following the same scheme is currently in progress. I would like to thank our collaborators from the Bergen University in Norway, in particular Dr. Nello Blaser, for initiating the collaboration and putting us in touch with the people who make the CYTOF experiments. And special mention also to my mentor, Benedict Einchank at the NIHS as well as NCI. And thanks to all of you for your attention, and please, if you happen to be in Madison, do not forget to visit my poster number 508 at the TransMed COSI. Thank you.